Hello everyone, welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of the cytoplasm, the important constituents of it and the various subcellular organelles such as the Golgi bodies, endoplasmic reticulum, mitochondria, ribosomes, lysosomes, peroxisomes, chloroplast and nucleus. This module would give us a fairly good idea about the ultrastructure of a cell and the functions of the various organelles present inside the cell. Structure and function of cytosol. The fluid component of the cytoplasm is the cytosol, while the metabolically active contents of the cytoplasm are the organelles. Apart from being metabolically active, organelles are permanent residents of the cell which would survive cell division. That is, they reappear in the daughter cells following cell division. The cytosol is a complex mixture of substances dissolved in water. Although water forms the large majority of the cytosol, its structure and properties within cells is not well understood. The concentrations of ions such as sodium and potassium are different in the cytosol than in the extracellular fluid. These differences in ion levels are important in processes such as osmoregulation and cell signaling. The cytosol also contains large amounts of macromolecules which can alter how molecules behave through macromolecular crowding. The entire contents of a eukaryotic cell within cell membrane minus the contents of the cell nucleus are referred to as the cytoplasm. In prokaryotes, most of the chemical reactions of metabolism takes place in the cytosol, while a few take place in membranes or in the periplasmic space. In eukaryotes, while many metabolic pathways still occur in the cytosol, others are contained within organelles. Organelles occur in two forms: freely within the cytosol or enclosed in the membrane. The cytoplasm also contains substances which are not metabolically active. These are called inclusion bodies. Inclusion bodies are the transitory residents of the cytoplasm not involved in cell metabolism and do not survive cell division. They comprise mainly accumulated metabolites, lipid droplets, pigments and minerals, and are sporadically distributed in body cells. The eukaryotic cell is enclosed in a limiting membrane called the plasma membrane or the plasmalemma. Cytoplasmic organelles. Organelles are the metabolically active and permanent residents of the cell. Organelles exist in two forms: membrane bound and not bound by membrane. The principal cytoplasmic organelles include mitochondrion, ribosome, Golgi bodies, endoplasmic reticulum, chloroplast, lysosomes, peroxisome, nucleus. Mitochondrion. Unlike most cytoplasmic organelles, mitochondria are large enough to be seen in the light microscope, and their presence within cells has been known over 100 years. As with most other organelles, mitochondria possess recognizable morphological characteristics while exhibiting considerable variability in appearance. The typical mitochondrion is sausage shaped, approximately 0.2 to 1 micrometer in cross sectional diameter and 1 to 4 micrometer in length. In some cells, such as those of early embryos, mitochondria are almost spherical in shape, whereas in others such as fibroblasts, they are elongate, thread-like structures. An average mammalian liver cell contains about 1500 mitochondria, corresponding to 15 to 20% of the cell's volume. Mitochondria are even more numerous in muscle cells, which require large amounts of ATP to fuel their contraction. A particular striking arrangement of mitochondria occurs in sperm cells, where they are located in the cell's mid-piece just behind the nucleus. The movements of the sperm are powered by ATP produced in these mitochondria. Mitochondria are also prominent in many plant cells where they are the primary suppliers of ATP in non-photosynthetic tissues as well as a source of ATP in photosynthetic leaf cells during periods of darkness. The internal structure of a mitochondria is as shown in the image. Each mitochondrion contains an outer membrane and a complex inner membrane system. The outer membrane completely encloses the mitochondrion, serving as its outer boundary. Although part of the inner membrane may lie just inside the outer membrane, much of it occurs as deep folds or invaginations called cristae. In some cells, as in those of the mammalian liver, the cristae are white sheets that across the entire diameter of the mitochondrion. In most plant cells, the cristae have a more tubular shape. The infoldings of the inner membrane greatly increase the amount of surface available to house the machinery needed for aerobic respiration. The membranes of the mitochondrion divide the organelle into two aqueous compartments, one within the center of the mitochondrion called the matrix and a second between the outer and inner membrane called the intermembrane space. The matrix has a gel-like consistency owing to the presence of a high concentration of water-soluble proteins. Mitochondrial membranes. The outer and inner membranes have very different properties. The outer membrane is composed of approximately 50% lipid by weight and contains a curious mixture of enzymes involved in such diverse activities as the oxidation of epinephrine, the degradation of tryptophan, and the elongation of fatty acids. In contrast, the inner membrane has a very high protein to lipid ratio, more than 3:1 by weight, which corresponds to approximately 1 protein molecule for every 15 phospholipids. At least 60 different polypeptides are found within the complex structure of this membrane. The inner membrane is virtually devoid of cholesterol and rich in an unusual phospholipid, cardiolipin. Both features are characteristic of bacterial plasma membranes, from which the inner mitochondrial membrane is presumably derived by evolution. The outer mitochondrial membrane is thought to be derived from an outer membrane found as part of the cell wall of certain bacterial cells. Both contain porins, integral proteins that form large non-selective membrane channels. Because of the porins, the outer membrane is especially permeable, allowing molecules up to about 10,000 Daltons to pass freely into the intermembrane space. The inner membrane is highly impermeable. Virtually all molecules and ions require special transporters situated in the inner membrane to gain entrance to the matrix. Mitochondrial matrix. In addition to various enzymes, the mitochondrial matrix also contains ribosomes. Mitochondria possess their own genetic material and the machinery to manufacture their own RNA and proteins. This non-chromosomal DNA is important because it encodes a small number of mitochondrial polypeptides that are integrated into the inner mitochondrial membrane together with polypeptides encoded by genes residing within the nucleus and imported from their site of synthesis in the cytosol. Functions of mitochondria. Mitochondria play a critical role in the generation of metabolic energy in eukaryotic cells. They are responsible for most of the useful energy derived from the breakdown of carbohydrates and fatty acids, which is converted to ATP by the process of oxidative phosphorylation. Most mitochondrial proteins are translated on free cytosolic ribosomes and imported into the organelle by specific targeting signals. In addition, mitochondria are unique among the cytoplasmic organelles already discussed in that they contain their own DNA, which encodes tRNAs, ribosomal RNAs, and some mitochondrial proteins. The assembly of mitochondria thus involves proteins encoded by their own genomes and translated within the organelle as well as proteins encoded by the nuclear genome and imported from the cytosol. The matrix contains the mitochondrial genetic system as well as the enzymes responsible for the central reactions of oxidative metabolism as shown in the image. The oxidative breakdown of glucose and fatty acids is the principal source of metabolic energy in animal cells. The initial stages of glucose metabolism or glycolysis occur in the cytosol where glucose is converted to pyruvate. Pyruvate is then transported into mitochondria where its complete oxidation to carbon dioxide yields the bulk of usable energy or ATP obtained from glucose metabolism. This involves the initial oxidation of pyruvate to acetyl-CoA, which is then broken down to carbon dioxide via the citric acid cycle. The oxidation of fatty acids also yields acetyl-CoA, which is similarly metabolized by the citric acid cycle in mitochondria. The enzymes of the citric acid cycle, which are located in the matrix of mitochondria, are central players in the oxidative breakdown of both carbohydrates and fatty acids. The oxidation of acetyl-CoA to carbon dioxide is coupled to the reduction of NAD+ and FAD to NADH and FADH2 respectively. Most of the energy derived from oxidative metabolism is then produced by the process of oxidative phosphorylation, which takes place in the inner mitochondrial membrane. The high energy electrons from NADH and FADH2 are transferred through a series of carriers in the membrane to molecular oxygen. The energy derived from these electron transfer reactions is converted to potential energy stored in a proton gradient across the membrane, which is then used to drive ATP synthesis. The inner mitochondrial membrane thus represents the principal site of ATP generation, and this critical role is reflected in its structure. First, its surface area is substantially increased by its folding into cristae. In addition, the inner mitochondrial membrane contains an unusually high percentage of proteins which are involved in oxidative phosphorylation as well as in the transport of metabolites between the cytosol and mitochondria. Otherwise, the inner membrane is impermeable to most ions and small molecules, a property critical to maintaining the proton gradient that drives oxidative phosphorylation. In contrast to the inner membrane, the outer mitochondrial membrane is freely permeable to small molecules. This is because it contains proteins called porins, which form channels that allow the free diffusion of molecules smaller than about 6,000 Daltons. The composition of the intermembrane space is therefore similar to the cytosol with respect to ions and small molecules. Consequently, the inner mitochondrial membrane is the functional barrier to the passage of small molecules between the cytosol and the matrix and maintains the proton gradient that drives oxidative phosphorylation. Role of mitochondria in the formation of ATP. Mitochondria are often described as miniature power plants. Like power plants, mitochondria extract energy from organic materials and store it in the form of electrical energy. More specifically, the energy extracted from substances is utilized to generate an ionic gradient across the inner mitochondrial membrane. Mitochondria utilize an ionic gradient across the inner membrane to drive numerous energy requiring activities, most notably the synthesis of ATP. When ATP formation is driven by energy that is released from electrons removed during substrate oxidation, the process is called oxidative phosphorylation. Oxidative phosphorylation can be contrasted to substrate level phosphorylation, in which ATP is formed directly by transfer of a phosphate group from a substrate molecule to ADP. Oxidation reduction potentials. The early work of Warburg and Kalin using spectrophotometric techniques led to the concept that cell oxidations are brought about by electron carriers arranged in a chain of oxidation reduction or redox potential. Since the movement of electrons generates a separation of charge, the affinity of substances for electrons can be measured by instruments that detect voltage. Protein import and mitochondria assembly. In contrast to the RNA components of the mitochondrial translation apparatus, most mitochondrial genomes do not encode the proteins required for DNA replication, transcription or translation. Instead, the genes that encode proteins required for the replication and expression of mitochondrial DNA are contained in the nucleus. In addition, the nucleus contains the genes that encode most of the mitochondrial proteins required for oxidative phosphorylation and all of the enzymes involved in mitochondrial metabolism. The proteins encoded by these genes are synthesized on free cytosolic ribosomes and imported into mitochondria as complete polypeptide chains. Because of the double membrane structure of mitochondria, the import of proteins is considerably more complicated than the transfer of a polypeptide across a single phospholipid bilayer. Proteins targeted to the matrix have to cross both the inner and outer mitochondrial membranes, while other proteins need to be sorted to distinct compartments within the organelle. The import of proteins to the matrix is the best understood aspect of mitochondrial protein sorting. Most proteins are targeted to mitochondria by amino terminal sequences of 20 to 35 amino acids that are removed by proteolytic cleavage following their import into the organelle. The pre-sequences of mitochondrial proteins, first characterized by Gottfried Schatz, contain multiple positively charged amino acid residues, usually in an amphipathic alpha helix. The first step in protein import is the binding of these pre-sequences to receptors on the surface of mitochondria. The polypeptide chains are then inserted into a protein complex that directs translocation across the outer membrane. The proteins are then transferred to a second protein complex in the inner membrane. Continuing protein translocation requires the electrochemical potential established across the inner mitochondrial membrane during electron transport. The transfer of high energy electrons from NADH and FADH2 to molecular oxygen is coupled to the transfer of protons from the mitochondrial matrix to the intermembrane space. Since protons are charged particles, this transfer establishes an electric potential across the inner membrane, with the matrix being negative. During protein import, this electric potential drives translocation of the positively charged pre-sequence. Proteins are targeted to the TOM complex in the mitochondrial outer membrane by amino terminal pre-sequences containing positively charged amino acids. The pre-sequence first binds to TOM20 and TOM22 and is then transferred to TOM5 and the import pore TOM40. Following passage through the outer membrane, the pre-sequence binds the intermembrane domain of TOM22 and is passed to the TIM23 complex in the inner membrane. In the matrix, an HSP70 chaperone associated with TIM44 acts as a ratchet using ATP hydrolysis to translocate the protein across the inner membrane. Let us now summarize what we have learned in this session. Mitochondria, which play a critical role in the generation of metabolic energy, are surrounded by a double membrane system. The matrix contains the enzymes of the citric acid cycle. The inner membrane contains protein complexes involved in electron transport and oxidative phosphorylation. In contrast to the inner membrane, the outer membrane is freely permeable to small molecules. Mitochondria contain their own genomes which encode rRNAs, tRNAs and some of the proteins that are involved in oxidative phosphorylation. Most mitochondrial proteins are encoded by the nuclear genome. These proteins are translated on free ribosomes and imported into mitochondria as complete polypeptide chains. Here are a few questions for you to work out. 1. Briefly explain the characteristics of cytosol and list out the various cytoplasmic organelles it possesses. 2. What two proteins of the mitochondrial inner membrane allow it to have unusually high metabolic activity? 3. What roles do molecular chaperones play in mitochondrial protein import? 4. Differentiate the characteristic features between the mitochondrial matrix and mitochondrial membrane. 5. Explain some of the important functions of mitochondria. Here are a few books for your reference. Biochemistry by Donald Voigt and Judith G. Voigt, John Wiley and Sons Incorporated. The Cell by Jeffrey M. Cooper, Robert E. Hausman, 3rd edition, ASM Press. Thank you for watching this session. Let's meet in the next session with another topic. Till then, bye.